Okay, good morning. So this is week 12 computer lab session. This is a second last session towards the end of the semester. So we will continue to use example to do manifold learning related topic. First, we will look at isomap method with an example. Okay. Then we will look at manifold learning or dimension reduction toolbox. So this toolbox contains a lot of manifold learning methods. Third, we will look at how can we use magic to do imputation. Okay. So the magic is based on this manifold concept to follow the global structure of this underlying structure of manifold formed by the cell with their profiles, and try to fill in those missing value and in the sparse single cell RNA-seq matrix. Then we will look at the tensor data type in MATLAB. And then there is a toolbox, and we can take a look. After that, I'm going to move back to the single cell gene expression, the interface. Today we will look at cell subsampling method. Okay. Subsampling means you will downsample your cell. If you have 1 million, you want to use only 500,000, and to reduce the burden of your computation, you want to have this catching approach to get those small set of cells and then look at the global pattern first very quickly. This becomes an issue when you have larger and larger data set. So once you finish this downsampling analysis with a small size subset of cells, then you have a global, a bigger picture of what the cells in your sample, what the structure, what do they look like. If you have two samples, whether there's strong difference or not, then you can move back to the whole set of data and use larger computer to do it. So we will look at a method called geo sketching. There's a paper published on that. And at last, we will go through some of these options in the toolbox, let you to get a complete functionality work through for all the functions available through the menus, the drop down. So toolbox 2. Okay. So I encourage you first update your toolbox. For example, the geometric sketching method I just implemented yesterday, just add in by calling a Python function. So if you don't have the latest version, you don't have this function in the toolbox. Okay, first, I will try Google Manifold Learning MATLAB. On my page, the first link is what we want to use. I don't know whether you get the same page or not, but this is one of the functions that we can directly copy the source code and play in MATLAB. Okay, so this is the page. Do you have this, everybody? Okay. So when you click this function, there's only one function here. So we can click the name of this function to show the source code. So this is a very small example to illustrate with the example. Okay. What you can do, you can copy the whole thing and then paste into MATLAB and to see the results. That's it. But what I want to do is just copy first this section to generate the example data, okay? And then copy the next section one after another. So I copy and paste this to generate this original data. So you can learn a lot from the source code and see how this data is generated, how this is plotted. So it generates three columns of points, okay? So X, Y, Z. X and Y follow cosine, sine as a function of t, time t as steps. And also Z, okay? Randomly put in the column. Then you generate this shape. So the color was indicated by the rank, the order. So if you have this point start from early in the X and Y position, then you have this dark red color and go over the spectrum to the end of dark blue. So this is coded by the order, where the order is given by the time t, okay? Sorted. So this is a way to generate this data. We use the color to keep tracking on the order of this data points. Then we move to the next section here. I think this part of the code is to generate a KNN network for the points. Okay, we can copy and paste here, and there's no figure, no other visualization available for this section. There's just a KNN network. It's not a K network, it's a distance matrix in the DT. Okay. So okay, so next section we copy. This is a kernel function. So if you recognize this, you transform the distance. So dt contains the distance between given points and 2 points. And then for each point, the distance we convert using this function to become the weight between the link for the link. So you can copy this. So in this example, there are 2048 points. And now we get this DO, which is a matrix that's 2048 by 2048. So this is a pairwise network. It's a Genesis matrix for the network. So then we apply this eigen operation and to get the projection of xy in 2D. And the result is saved in the V vector. Okay, so you can copy this at the end to finish this embedding. Okay, on your left panel is original data, and on your right, you see, okay, this is from 3D to 2D and keep this Swiss roll shape unwrapped to allow you to look at the relationship between, for example, the dark red points to the dark blue points. Okay, they are the distance from here to here is maximized. Okay, unwrapped. You cannot do this, use PCA. PCA will put a 2D like this. If you look at this 2D and something like that, PCA will end up with it at this moment. Okay. So we finished the first example, and by close all, clean all, our second is to look at this manifold learning toolbox. So you may want to search for the keywords, MATLAB toolbox dimensionality reduction. That's the toolbox we are going to use. So this is the author. He is the author for the Tisney method algorithm, I believe. So he developed this toolbox contains this method related to the dimensionality reduction. Okay, so isomap is here, Laplacian eigenmap in the number 10 here in the list. Kernel PCA, diffusion maps. So, and Tisney is in the position 31. I don't know if he get this. U map is not here. Okay, let's see how can we install this so we can try some examples provided in this toolbox. What we need to do is just click this download. So click here to download your toolbox and then show it in the folder of download. So let me get rid of others. So that's in my download folder. And I can right click, use 7-zip to extract here. Okay, so this is the first unzip this GZ file into this table. Okay, I can delete the original. Then run 7-zip again to extract here. And then I can delete the zipped file. I only have this folder here. So that's the place which we can point our MATLAB into this folder, and then we are ready to go. So download the package, unzip until you have this folder, and then copy the folder name, and then to do the CD here, to change the folder, okay, to there. Okay, so you see my folder, the working folder right now is in the downloads and this DR toolbox, and I can see all the functions inside here. So I will try this DR GUI. So this is a graphic user interface. I just right click and run it, see whether two. I see. So we need add these two folders into the search path. Okay, you select these two because all the functions are under these two folders. So you select them to add the path for the selected folder. And then run this GUI again. We should have this interface. Okay. All the major functions are inside these two folders to allow this program to the environment to access the function inside. We have to add these two folders into the search path. Then you come back again to run this DR GUI. You have this interface, and then we can play with loading example data and select a different method. Just play around. Okay. Yeah, so this is very common way is you generate, you download the folder. Oh, you're not in there. Do you know where you download?
Here.
Oh, yes, yeah. Yeah, so let's right click this and unzip it.
Okay.
Yeah, again, so extract here. Okay, now go into here, unzip this. Okay, go into here. Yes, that's the place to copy the whole folder.
Oh, this folder. Okay.
Yeah, CD to here. Yep.
Okay.
They're in there, and then you have to add these two parts. Move down, select these two folder, select and then right click. Add the parts, select folder, and do the same for another folder.
For this one as well?
Yeah,
okay.
We need both.
Okay.
Yeah,
okay,
I think now you installed it, and then yeah, just play this. This provide very good. Yes. Do you know where you installed?
Yeah.
Yeah.
Some download check right
Yeah, to finish this string by another single quotation. Okay. Yeah, yeah. And then move down, add this two folder to the parse. Right click. Oh, not inside. Yeah. Yes. Yeah. Add two parse. Go down. Yeah. Select it, and do the same thing for another folder. Yeah, now you should be able to type DRGUI. GUI GUI. Yeah. Yeah, so you can try it. Play this. Not working yet. In there. Okay. Okay, it works. Yeah. Yeah, so you can play with the example the toolbox provided. Okay, yes, good. So the first example data is a Swiss roll. We select it and we click scatter plot. Something wrong with this. Estimate the dimension using those different methods. Computing the map. And here you select different method, Laplacian. Yeah, I found there problem with I see error in here. What's yeah, there's some problem with this interface, some function. Maybe it worked in the old version of MATLAB, but not now. Have some problem in. So I don't know. Let's try another data, maybe this. Still. Do the dimension. yeah, somehow you can see the results. So you embed this into a 2D like that. Mhm. Oh. Manifold chart. Okay, so this toolbox gave you a lot of options. The function is inside the techniques. So you see all this implementation of the method mentioned in the introduction of the web page. I think the author is using a Mac machine, and I don't know why there are so many this kind of temporary files, which we can really just delete them. A lot of junk stuff here. Yeah. Okay, so if you want to use this, you may have to find a way to load your own file and your own data in there, rather than just use this example files. And there are two different ways. One is to read from Excel dataset, and then also you can load from MAT file or TXT or tab text files. Okay. 3D cluster. The other is just example data. Okay. Any questions? No. We are going to move to our next task here to learn how to use magic to do the imputation. Okay, let's clean this. Close all. Okay. So remember, your current working folder is in this DR toolbox. So we are going to move back to our original. So let's select the place. Okay, back to here. If you click this, you have this previous path or previous working folder. You select the right one. We come back to the place where your default working folder here, and where we have the toolbox installed. Now we are going to use example data and export it into the workspace and see how can we use magic to do the imputation. So let's get the example data, okay? You see, from those menus, we don't have imputation method because I don't suggest you to use. But in some case, if you need imputation, I show you how to do it from the command line. Okay. So now we export the data to the workspace. Now we can export this SCE, which is the whole object, contain everything, and also we can export the expression matrix into the big X, and also you have a gene name or embedding of the position of the cells. So I just exported this three, and now you have a workspace. You can see this original UMI matrix. So now I want to impute my X by calling magic. Magic will help you to change the value of zeros into something else based on the manifold structure of all the cells relationship. What we can do is to do X the output imputed. Okay, it's a variable name. Okay, then we can run, and here you can have magic. And then put your X in there. So that's it. So if you have this toolbox installed properly, and you should be able to run this external function magic, and then press. Okay, this method is super fast as well. We talked about this principle, the pipeline, during the lecture. So it starts with the distance between cells, then the kernel function to change the distance into affinity, and then there's soft thresholding, and then cut some of the links. And then they used random walk. And then at the end, they impute all the values. So from this function, you get the imputed version of X, you see the dimensionality is the same. Okay, same number of genes, same number of cells. But if you double click, look at the values filled in, okay, compared to your original X, this imputed version looks like this. Okay, so now if you try to do a scatter plot between two genes, for example, the gene number 1, remember in our case, the rows are the gene, and the X gene number 10, for example. In this scatter plot, you look at two genes correlation. Okay, let's close this figure. Okay, don't see anything here. So in your x-axis is expression for the first gene. In your y-axis is expression of the number 10 gene. Okay. But if you use imputed version of look at it. All right, you get a very different picture. Yeah, much higher, of course, yes. So now it looks like these two genes are positive correlated. But if you do this correlation analysis from the original data, because of the zeros, you'll never be able to recover any of the correlation. Okay, so let's see what's the name of the two genes. The gene 1 is here, so I will label X label gene number 1. Okay. You see, this is a MRPL15, and you can do the Y label. Our example is gene number 10. Okay, you get this picture. It's based on the imputation results. These two genes seems positive correlated. So you can play with this for other pairs of genes. You can select number 100 against 110, whatever gene in there. I think this is one of the applications I can think about why we want to use imputation in the single cell data. You try to find those undetected correlation between gene co-expression. Get a similar picture, same picture. Okay. Yeah, it's good. what I found in some case, these two genes are not monotonically increase or decrease. The relationship is nonlinear. Sometimes you have a shape like this. Okay, it was positively correlated and then become negative. So this method probably can help you to discover some nonlinear correlation between genes. So this is another pair, and it's very different. They always have a very different, depends on which two genes you look at. So what to do with this kind of information? So you may want to add in the third gene's expression. So it maybe this group of cells expression level is high in the third gene. Okay. In that case, what we can do is to do the 3D. Okay, let's do it scatter 3. Then you put the third gene in this just random gene names, gene IDs. Okay. And then I can use X label in 327, Y label 1299. Okay, so this plot help you to understand the interplay between 3 genes and the shape of this 3 genes relationship defined by the cell samples. So depends on how many points you have. So this method won't be able to do it in the large scale. You have to know which 3 genes you want to check. Because if you do this, n choose 3, you have a large number of possibilities. You'll never be able to look at them one by one, but you have to know which 3 genes you want to look at. Okay. Yes. Yeah, so this is not exactly not the expression level, it's a transformed value. So yeah, don't take that into as an expression value, because expression shouldn't be greater than smaller than 0. Right? Yeah.
Based on the gene name to find the data, the expression data.
Oh, if you want to know the name, so you can do what I want to do is you can get. So for example, gene, you put a name in there. R, for example, then you found. So that this will give you an ID. If you don't have this gene, then you have 0, okay, nothing written. Again, as I said, this function hasn't been built into the toolbox through the interface because I don't encourage you to use imputation. You don't know whether the imputation step distort the relationship or not. But if you have a strong biological background, believe these 3 genes should have some correlation, and you can use the imputated values to test to provide some third party of this evidence. Because you installed the toolbox, after you click the run with a dot and use the tab key, you found a lot of these functions, which I call them external functions. So we can call some of those functions provide through the interface, some are not. But if you check, you probably want to expand this functionality, and you by yourself you can select some of those. Okay. For example, T-scan is for pseudo-time. Okay, pseudo-time analysis. UMAP you can call from here. There are 3 different versions of UMAP. UCell. Okay. Okay, so next I'm going, so we finished. Okay, so next time I'm going to show you how we can create the tensors in MATLAB, and then we take a break. Now, so far what you can get is like a big X matrix. You can get the values, the position 3 and 4, and you get, okay, the expression is 1, but in the imputated version, 3 and 4 is 0.26. So all these data sets are in the either vector or matrix form. So now you can also define a variable, say y. You can give this position 2 and 3 and also 5. Okay, assign this into value 1. This create a data, okay, the variable y. Its dimensionality is 2 by 3 by 5. So now it becomes a tensor. Let's make another example, and this time I'm going to create a random tensor. Okay, say Z. Now I'm going to use 16 by 16, 9, okay. Make it larger, maybe 25. Okay, so you just gave one more additional subscript as an index, and you generate a 25 by 25 by 9 random numbers and filled in this Z. It's not a matrix, it's a three-way array, okay, it's a tensor. You can go beyond. See, I add another one that's fourth or next 3D for even higher, okay? MATLAB should be able to handle this. So this data is random generated data. If you have your own high dimensional spreadsheet, okay, you can put first spreadsheet is 25 by 25 in the first layer. And then you have another, and to take the whole set of the data points, arrange them in this 3D structure. So how can we extract the information from there? So for example, I want to extract the first, and this is extract the first layer of this 25 by 25 matrix. Okay. And then you can do the second. So we are extracting the data as a matrix from different layers by the index of a third position of this index. Okay, we can do this for 9 of them. Something like that, okay? You can also extract from other positions, for example, Z, and I want to the first position and the 12th position in the third, and the 5 positions. So you get the value from this qubit, one of position in there, okay? In this case, you extract the whole column, whole row, whole column, and layers. You can extract the third, okay? So this index is, you can use to extract the whole, I would say, column or whole slides or whatever, the way you define them. So you see, we can use this function to image SC, so pseudo color basically. To plot the use the color map to show the values in each of the position in this 25 by 25 matrix. So you can repeat this 9 times to see the whole tensor of the data. Okay, so I'm going to do this. Okay. 9, so I'm going to do, and I use subplot. I will define a 3 by 3 subplot each time I get one of this image as C Z K. I use this to generate My Z is 25 by 25 by 9. Each time I select one layers of the matrix, is plot them and put in this 3 by 3 subplots. Okay, at this moment, we can take 5 minutes break. Okay, so we are going to do some analysis with this tensor. Okay, so this small example, 25 by 25 by 9. So we will treat this as a single variable entity to give to function. But the majority of the function in MATLAB is designed for matrices. So if you want to do eigen decomposition, something like that, you will get error message. For example, if you do SVD and for this is Z, okay, SVD will say, okay, I can handle all type of these matrices, but not tensor. So we need to use other external toolbox that we will download Tensor toolbox, see whether we can use some of this function to do decomposition, okay? So let's first, I'm going to reconstruct this because right now it's meaningless to analyze this because everything is just white noise, random noise. What I want to do is I create some artificial structure among these different layers of data. So that's what I'm going to do. I'm going to loop through 1 to 9. I will change the position. So 25. So I have 1 to 15 position, 1 to 15 position for each layer gave by its value. Okay. So I changed the K. Let's do a for the subplot again. K to subplot image. So the value in the first layer, this yellow box, the value is 1, and this is 2, this is 3, 4, 5, 6. Okay. Is the all relative position. So it always gives this highest value as yellow. Okay. So if you switch on this color bar here, you can see the highest value in each subpanel changed from 1, 2, 3, 4. That's what we created. Okay. So once we have the tensor, basically we can do nothing about it if you use the native MATLAB environment. That's why we need to Google and search for this toolbox. This toolbox will create a tensor object, and a lot of function will be provided. What we can do is to download the source code. Again, this is in your download folder. You have to unzip it using extract all. Put into this folder, and you need to know where you unzip the folder. And then we need to add that parse into MATLAB. And after unzip it, the toolbox itself is inside my downloads folder, and then toolbox, there's two levels of the same name, but I have copied the whole thing in the copy. And you can start to see the function within this toolbox, okay? And come back to our MATLAB environment. You have to do is to add parse and give the string to this add parse. You can also do to find the parse and add to the environment. Okay, make sure you download and zip the file, then add the folder, the right folder here. So if you install the package correctly, you should have this function called Tensor. And if you give Tensor the Z, remember Z is a 3D array, and then it will return this object. I call it TZ. I know it's a tensor from the original 3D matrix. So once you have that, once you have the TZ, there are a lot of functions in the toolbox you can apply to this TZ. That's good. Working? Okay, great. Oh, you haven't installed it correctly because you yeah, so that's in your zip. You haven't unzipped the file. Yeah.
Okay. Yes, sir. I'll go ahead.
No zip. I think you are right.
It's the wrong
Yeah.
folder. Okay. So,
Yeah.
Yeah, I had unzipped the book.
Yeah. So that's right.
Okay. Thank you.
Yeah. Just yes, it should work. That's great.
Okay.
Yeah.
Thank you.
Questions? Okay, so this is an essential toolbox. If you want to do the tensor analysis, you have to have it because the official MATLAB, native MATLAB, does not support a lot of those. And once you have tensor, there are different type of tensor, and we just create a general version, so we are okay. Then what you can do is to do this CP decomposition. That's a major application. So this use alternating least square algorithm to do the CP decomposition to learn the major components in your 3D object. So we are going to use a function called CP underscore ALS. Okay. They give this to the TZ to this function, and it will give you the output. We need to know the value of K. Let's see. So I'm using this method. So we need to know the parameters. I think I missed the number of them. Number of K. Okay, so to use this, you need to give, like, I want to use the first 3 components. So you already have X, which is our object Z, TZ. Then I give 3. Okay, I just learn the top 3 components. So in the M is contains those components. Okay. Now I'm using the learned components to reconstruct Z. Okay, let's see, call it ZM. Okay, let's reconstruct it. And then ZM. Okay. So through this operation, I use decomposed and reconstruct this object, and then to get the data. And the data is the same as our original. So the original is 25 by 25 by 9, and this reconstruct is also 25 by 9. Okay. So this will finish the whole loop. We decompose the major components and use top 3 and multiplicate and reconstruct this original version of Z. Okay, original version is Z, and now is Z data. So what we can do is let's close. I don't want to close this. What I can do? Let's have a figure, and then for K, 1 to 9. I'm going to still do the subplot and use image and see. Now this time I use reconstructed data and see what we can get. Okay. So this is the, make sure you use a figure and to pop up a new window. So okay, so on this new figure pop up, this is my reconstructed version of my original matrix. You see some of these denoise effects. Okay, it was like that now here. You can still highlight each of these subplot and use the bar, the color bar to see the top levels. Okay. Okay, so this reconstructed data is the denoised version, but the major trend is also captured. So this block is 1, this is 2, this is 3, okay? Because we intentionally constructed something like this to give some information from the original data. And now the effect is to remove all the noise, but make this major pattern even more pronounced. That's cool. Okay. Yeah. It's a kind of different. Okay, you got a different version of it. Oh, you're wider. Yeah. Yeah, I think when you construct the Z, you use a slightly different one. Yeah. Which step? Let's find your place where you you add this 15. Okay, yeah, this is correct.
Okay, I will figure it out.
No problem. Yeah. Okay, everybody, we finish with the tensor analysis. So we are done with this. Okay. Now you can Google by search a paper like this and read this brief summary of this and to get some idea what they're trying to do. So this is a subsampling method. Imagine you have a large data set with several million of data and you have a different cell type. There's one big cluster, there are many cells. You don't want to subsample a lot from this uninformative cloud. Rather, you want to select not uniformly across this cell samples. You want to sample relatively more cells from this cluster, for example, in order to keep the real cell type in your subsampled data. You want to sample less from this group of cell, right? Eventually, you want to get this representative, much sparse data points, still reflect major components, major clusters in your original data, large data set. So the technique they use is called geometric sketch, and they provide this planned covering first, and then based on this covering, the sample depends on the volume of the cells in there, and to sample less from this dense region, sample more from this relatively sparse region. At the end, to achieve this geometric scaling of the original large data set, make it more representative for the downstream fast process of the original data. So this analysis may take several days, but you can have a very quick sketching, small data set to achieve major steps. So you have a quick idea of what's in your original data. Okay? So that's the goal. All right. So now if you search GeoSketch, then you come to this GitHub repository link. That's where they provide the source code developed in Python. And you can find how you install the package. Okay, this is not MATLAB. You have to come back to the mini conda. Okay. Again, this is a high advanced technology. You call from MATLAB, send the data to Python, and the Python process it, send the data back. So in this place, we do the pipe install GeoSketch. Okay. We have to install this before we can call the function from MATLAB. So let me know once you finish this step. Okay, good. Question. Okay, it's not finished. You don't have a mini code. Yeah. You can go to the website as mini codon to download and quickly install. Yeah, just search. Yeah. You have Windows installer. Yes. Let me see. You want to try maybe version. Yes, this should be fine. You just install this, and then you should have that black window. Mm-hmm. Then in that black window, you just type in the PIP pipe and then install that. Then we are done. Mm-hmm. Once you install that, we can come back to the MATLAB and close all this previous steps, clear all. I'm going to use example data to do this subsampling. For the first time user, you want to check your Python environment by click this to make sure your program can find where the Python comment is. So I know I installed my Python in this folder, so I know where the python.exe is. If you don't have this, you may want to select by use another, you select where your Python is installed. So once we have that information, we are ready to go. And also you installed the GeoSketch package already, so we can use experimental and here subsample 50% of cells. Okay. This function will allow you to sample half of the cell. You can repeat this several times until you get a small number of cells you want to work on. Okay, that's it. Do you know where you install your Python? I don't think so. It's in your user folder. Let's see, in your user, found your name, and then let's see where. Let me check what where. Yeah, so let's see where you install this. Do a dir, see which folder this is. Can you type here which Python? Which? That I don't know where you installed. Oh, let me check. So you have this. Then I can look at this properties. So I know, so you are installed here. That's where the folder is. Yes, I think that's your folder. Wait. Copy this. Yeah, paste to here. That's where you install. It says so your phone. Select this, yes. You are using old folder, old file. You haven't updated. You need to update this by this is your folder. Okay, you need to upload it by unzip. Just want to use the previous. You do this. So after that, you should be able to. This is just install the new version. So you click this and see, okay, this function will subsample 50% of cells. Continue, and then you, this is a purely random sample. If you want to just ignore any density of whatever, but you can use this geometric sketching method. Let's see, yes. Oh, the problem, too many, okay. It's error message. Yeah, so let me go back to fix this bug. So I believe this is because PCA I pointed to, oh, that's a confusion because my the PCA. Let's get rid of this this one here. Let me delete in the download folder. I delete this so the program don't get confused. Delete all this because now the PCA is pointed to another file called the PCA. We just so now after get rid of this, we should be able to go. Okay. In the set parse, remove this two. Okay. Okay, so after you remove that conflicting PCA function, you should be able to run this. So the sample was about more than 2,000 cells. Now it's only 1,000. You can repeat this into subsample cells. Okay, you know how to remove that toolbox link? You use old version, so you need to update here. Yeah, yeah, you don't have. So let me show you how to update. Nothing here. So make sure you're in the right position here. You have unzipped UNZ. I remember you should have this. Then just, this should be downloading. Then the toolbox should be here. Yeah. Working. No,
Mac.
yeah, Mac. Mac has problem. Nothing happened. I think it's right. It's working. What's error message then?
Users from here sketch.
You haven't installed this. Mhm. You probably have two different Python environment on your machine. Maybe, yeah. You don't know which one you installed for. In Mac.
The tool tries to look for a exe file. For Python.
Oh, that's a problem.
It doesn't find an exe file because
No, that's right. So how should I fix that? If the Mac machine, I should look for without exe. Yeah. Yeah. Okay, so far this has some problem within the Mac machine. Yeah. Okay, sorry. Still not there? Okay. Okay, let me show you. So you installed this here, right? Let's check your parse. So you MATLAB. Also point. So let's first remove those useless. Just make sure we know what we. Now if you do, it's not there. So now you add this into the parse. Okay. Now you should have this. I hope this is a new version. It's still old. This is still old version. You have done this. You are not able. Delete this. Anyway, let's unzip it. Oh, sorry, this is a different one. Yeah, so that's visual. So we keep updating this one. yeah, yeah, you have to find the correct one. You just in your history, in your command line history, you should have that. Just found spatial. I think that's one. Yes. Yeah, I think that should work after that. So next week we will look at those network construction functions. Okay. I gave you some examples here. For example, we can construct gene regulatory network by select some of the genes. This is a small network. Okay, by click this. Have you seen this before? Okay, yes. Okay. And you can compare across different IDs, batch IDs, and select genes you want to build into the network. Okay. To compare them side by side. Those are the random selected genes, so it does not make any sense. They shouldn't see much different. You shouldn't see much difference between two samples. I updated the function here, say on all markers. So I realized in Surate R package, people use this a lot to compare between different clusters to one cluster against all others to find a differentially expressed gene, or you call the markers for this cluster. Then repeat this for another cluster against, so you get a table. So I provide this function, and you can see, okay, I want to find for different cell type, and I will compute for each cell type and what's the marker genes or differentially expressed genes for this cell type against all others. So you get a table at the end. Normally, this will be a huge table. You can either save it or just play with this. So this table, first you have a group. Okay, that's the different clusters. You select those are the genes expressed highly in this group. So you can use those ones as a top marker genes. After that, you move to another cell type. Okay, so a Sina cell and then your alpha cell start from the top ones. This sorted by the p-value, adjusted p-value. And some cell type have more those D genes. And then you have B cell, those are the top ones for the B cell, beta cell, so which one is top? Okay, so this table will be the same as you use R package to generate. So that's an update for this function here. Okay. So now I use this example, come back to its original form rather than subsampled. Here we still have 2075 cells. Okay, I show you, I will show one of the function is called plot cell trajectory. That function will help you to construct a principal curve that will go through all the cells. It doesn't make sense for this example, but I just show you, you can do it by select trajectory and then you select spline fit. Okay, in here you get a curve. This curve goes through all the cells. Okay. So the purpose of this is try to label each cell a pseudo time value. It doesn't apply to this case because we have a different cell type. So how do you jump one cell type to another? So in which situation you can use this? We can, for example, extract. I will extract part of the cells. Okay. I will highlight cells in this group, and then select, delete. I will delete all the other cells unselected. So now I can use this spline fate or trajectory, because this is the cells arranged in a shape that suppose you have some sort of this pseudo time. So now I build a curve and assign each cell a value, okay? And then I can select some of the genes. There's some problem, I will fix that later. So you will find some genes. Expression will change from the start to the end, some feature genes, okay? If you believe the shape of this cluster of this group cell contain the information from one stage to another, then you can use this analysis very quickly to get those genes change from one end to another. Okay, it depends on your application. So right now, this is not a good example. Okay, this is not a good example to do that. But I just show you the possibilities there. So if you have an elongated shape of the cell cluster, and you believe from one end to another, there some of the development change is a function of the time. You can use this, create this artificial curve. Okay, then assign each cell a value, depends on where they are on the curve, and then start to find the gene change across the time. So I showed you we can get these O markers for different clusters, different cell types. For example, I will copy several genes from here. And then the function I want to show you is this called violin plot. Now you can select violin plot as a cell type as a group. Then you put some genes in there. Okay, the gene I selected are the examples. Then you will you can normalize it, and C is a good option. And you get those violin plot for marker genes. Okay. So the gene I selected are the highly expressed in the acina cell. So you see those kind of plots. That's for the publication. We have a lot of this in other people's publication, right? I don't believe there's a good way, but this option is there. So if you like to use this, you can export this to PowerPoint to show, okay, this is marker genes for acina cell. All right, I think that's all we can share today, and let me know if you have any questions, okay? By the way, I don't like the color, so you can provide this function to become black and white, okay? For the homework, I mean, the last time's homework about this classifiers you want to create for this provided big data spreadsheet. I don't want to spend time in lecture here to explain, but I do have a video from last semester. I will send you a link. You can check. So in the video, how we solve the problem, what's the major steps. Okay. If you want to know so-called the correct answer of your the task of your homework. For the last homework is a highly variable gene applications. You can work on your own cell type you like. Okay. Yongjian won't be here next week, but he will come back after next week. Okay. If no questions, I'm going to stop my recording. Yes. Oh, the final exams. Final exams is, I remember. Yeah, so I will send you a link. This is a Google form. There are 30 questions. Each question have 4 options. There's only one is correct. So you just select. It's open book, so you have all the slides on the internet. You can search. Okay. You don't have to come to here. You can in your dorm, in your airport, wherever you are, as long as you spend that 45 minutes. 45 minutes for 30 questions. Okay, once you submit that, I will collect those answers and compare with the standard answer. If everybody answered wrong for some questions, I will take that question out, because that means I made a mistake maybe. Okay. But anyway, we should have a way to evaluate and give you a score. You can look at those PowerPoint slides and yeah, to know where if I have a question, where I can find the answer. So in 45 minutes for 30 questions, you need to have a way to very quickly access your original, the place where the question is located. Okay. I don't think it's very hard.